and a coordinator of this webinar series, with Amy Stone of the Ohio State University Extension and Jody Ellis of Purdue University. Today's presentation looks at the ecological impacts of an area after emerald ash borer has come through it. This is an ongoing research project that is being studied in part by today's presenters, Wendy Klooster and Kevin Rice, who are both graduate students at The Ohio State University. Wendy is a PhD candidate and is working with John Cardina in the Horticulture and Crop Science Department, Ohio State. Her focus is on the ecological impacts of EAB and how it affects the establishment and spread of non-native invasive plants. She received her master's degree in horticulture from Michigan State University. Kevin is also a PhD candidate studying the ecological effects of EAB in Dan Herm's laboratory at Ohio State. He received a bachelor's degree at the University of North Carolina at Asheville and a master's degree from Auburn University, where he studied the effects of an invasive ant on native arthropods in natural settings. Before coming to Ohio State, he worked as an extension agent at the University of Arizona. I wanted to let you know that continuing education credits will be available for this session, and we will give you specifics as to acquiring those credits at the end of the presentation. If you need verification that you attended this webinar for other purposes or for self-reporting, please indicate that when you email your request. I wanted to let you know that you can feel free to type your questions and comments in the chat feature on the webinar screen. Your questions will be written down. To keep the presentation moving, our presenters will be answering any questions after the presentation. This webinar is being recorded and will be available for viewing later today. Go to www.emeraldashborer.info to find the URL for the recorded webinar. Thank you very much for your interest today, and we hope that you will feel free to email us with your thoughts about today's presentation. We always like to hear if these sessions are helpful and any other input you'd like to offer us. And with that, welcome Wendy and Kevin. And Wendy, feel free to start your presentation.
Great, thank you, Robin. I hope everyone can hear me just fine. So today I'll be talking about what happens after EAB. And again, my name is Wendy Klooster. I'm also working with Kathy Herms, John Cardina, Dan Herms, Kathleen Knight, and Deb McCullough. All participated in the research that went into this talk. So just for a little background, this is a graph that came out of a publication from Kamal Gandhi and Dan Herms looking at the cascading effects of an alien insect herbivore. And one of the pathways that they show is the alien insect herbivore can cause tree mortality, which causes gap formation, which can alter ecological interactions and the successional trajectories. So just keep that in mind. That's kind of what I will be talking about today. And the alien insect herbivore in particular is the emerald ash borer, which is an exotic beetle that came over from Asia. It was discovered in southeastern Michigan in 2002 and is now present in 15 states and 2 Canadian provinces. The emerald ash borer larvae feed on the ash phloem, which causes tree mortality. And already there are tens of millions of ash trees dead, and mortality approaches 100% within 4 to 5 years of infestation. And there are currently no successful eradication or control methods. There are some that can delay mortality, but nothing that is effective completely. Ash mortality, as I mentioned, results in canopy gaps, which can alter the microenvironment of the understory, including light, temperature, and moisture, and this can have an impact on the understory plants. So what I'm looking at in particular is what is going to fill these gaps. So I'm asking, will ash reestablish? Will other native species take over in the gaps, or will invasive species fill in those areas? So first thing I looked at is the fate of ash seeds. So part of looking at if ash will regenerate is looking at if the ash seeds will persist in the seed bank longer than the EAB persists in the forest. So my study takes place in southeastern Michigan, and also we're collaborating with Kathleen Knight in western Ohio. In Michigan, we're working in 7 state or metro parks within the Huron River watershed. And in Ohio, there are 7 metro parks, 12 state lands, and 12 private lands. So the plots are classified according to the dominant ash species. We have white ash for the xeric sites, green ash are considered mesic sites, and black ash are hydric sites. We've also established some plots without canopy ash, which we are considering kind of a non-ash control. So for the methods, we've started taking, beginning in 2005, we started taking ash mortality ratings, which is on a scale of 1 to 5, 1 being healthy ash tree with no sign of decline, and 5 is completely dead, and 2 to 4 represent continuing stages of decline. So this is being done in Michigan and Ohio sites. In Michigan, we also have collected seed bank samples, and those were collected in 2005 to 2008 each summer. We sampled over 80,000 centimeter cubed of soil, which is approximately 22 gallons. And we used those samples to check for ash seeds and their viability. So we did this in 2 different methods, collaborating with Deb McCullough's lab at Michigan State. We did pressure and tetrazoleum analyses and germination testing at OSU. So we also conducted seedling and sapling counts in Michigan and Ohio. We classified the trees according to their size, with new being the new seedlings with cotyledons, established less than a foot tall, established over a foot tall, trees that are over breast height but less than 1 inch diameter, trees over 1 inch diameter, and also we've been looking at shrubs of native and invasive species that are over 1 meter tall. Here's a figure that shows the results from the mortality in Michigan. And as you can see, beginning in 2005, mortality was approximately 50% throughout the area. But by 2007 and on, it reached nearly 100%. And it's still floating around that 100%, 99% level. Just as new saplings enter the size class, they become living trees in our category. This graph looks at our results from the seed bank sampling, and this is just looking at the pressure and tetrazoleum testing at Michigan State. So you can see it's done by year, and we've also separated it by the hydro class, which is again in relation to the dominant ash species. And you can see each year the number of intact seeds, the number of fragments, and the number of viable seeds. And the number of intact seeds was 52 in the first year, and then it decreased to 1 in 2007 and 0 in 2008. And the number of viable seeds decreases from 24 in 2005 to 0 in 2007 and 2008. So you can see that there was a pretty drastic drop in the number of viable ash seeds and even seeds present. So looking at the results from the germination testing at OSU, we also found intact seeds in 2005 and 2006, some of which were viable, but nothing in 2007 or 2008. So just to summarize, in 2005 we sampled 27 plots at 2 depths, and we found a total of 63 intact seeds, 35 of which were viable for both methods. In 2007, we doubled the number of plots to 54, but we only took 1 layer of sampling, and we found 40 intact seeds, 13 of which were viable. And by 2007, we had only 1 intact seed which was not viable, and no intact or viable seeds in 2008. So looking at the seedling and saplings, we had, you can see on this graph here, we have very few new germinates. These are again the ones with cotyledons in 2009, but we have a large number, and this is on a log scale, so just keep that in mind, the scale is log. And there's quite a few seedlings that are less than a foot tall, and then it declines up to over breast height, which is due to natural self-thinning. And this is the same type of graph, but showing for 2010, and you can see that it's the same general trend, but there's even fewer new seedlings found with cotyledons. So it just shows that there's really nothing more germinating from the seed bank. And this is data from Ohio. Now the thing with Ohio is that the plots span the ash canopy mortality. So there are some plots that are still with healthy ash trees, and then others that are already at the level 5 with all of the ash trees dead. So you can see how this graph shows the relation between the number of seedlings and the ash canopy rating. And so here you see plots with ash canopy ratings of 1, healthy ash, have a fair number of ash seedlings, and once you get to the plots that are later stages of decline and dead, you don't see any more seedlings. And this is in 2008, the numbers are low, but then even in 2009, after a mast year where the numbers are higher for the 1 through 4 sites, even still in the number 5 sites, there's very few new ash seedlings germinating. So to summarize, in Michigan, the plots now exceed 99% ash mortality, and we're finding no evidence of a persistent seed bank and little to no new ash germination. In the Ohio sites, where they span a range of 0 to 99% mortality, we're finding little to no new germination in the Category 5 sites. So it seems that EAB persists longer than the ash seed bank, and we will have to wait to see if the seedlings and saplings that are already established will be able to survive. And it just seems that unless the EAB population is eliminated, that they have a potential to threaten all ash in eastern North America. So now that we have an idea of what what is happening with ash, we're looking at the possible threat of invasive species coming in to fill these canopy gaps. So here's a few of the invasive species that I'm looking at. We have barberry, honeysuckle, and the bittersweet. And just to give an idea, the sources of invasive plants are either they can be shade-suppressed juveniles that are already present in these sites and have become released with the canopy gaps. Also, you could have seeds that are newly dispersed from individual plants in the surrounding landscape, or you could have new seedlings emerging from the seed bank. But for today's talk, I'll just be focusing on the first two, the shade-suppressed juveniles that are already present, and possibly the newly dispersed seedlings. So here's all of the invasive species that I'm looking at. We have barberry, celastrus, autumn olive, euonymus, privet, multiflora rose, the honeysuckles, the vining and the shrub honeysuckles, and the common and glossy buckthorns. So one of the things that we're looking at is to quantify the canopy gap fraction in relation to the ash mortality. So we have taken hemispherical photographs in multiple points throughout each plot, so 5 to 20 points per plot, and we're looking at the openness in relation to the ash mortality and also to quantify the growth of the understory plants. So to find the understory plants, we've done a little survey and we've tagged and measured an invasive species. As you can see in this picture here is the invasive species, which is a buckthorn. And you can see the native species that we've paired with it. And so we're measuring the growth rates of the native and the invasive to kind of look at how they relate to the canopy gap fraction. So we have the length by width by height, and we've remeasured them over the years. So we have the relative growth rate, which is the final volume minus the initial volume over the initial volume. And we've also, again, done the seedling and sapling counts for the native and invasive species, the new, established, and shrubs that are over a foot tall, or I'm sorry, over a meter tall, and the trees. And looking at some of the results from the canopy gap, this is just a subset of data as we have analyzed some of the pictures already. You can see that in 2009, looking at it by hydroclass, hydric, mesic, xeric, and then also our non-ash plots, there's a pretty wide range of the minimum to maximum percent gap fraction. And you can see how it's somewhat different between the different hydroclasses, but the medians are still relatively similar. And looking down to Highland, this is just one of the parks that we're working in with multiple plots. You can kind of see the difference between 2009 and 2010, where it almost looks like the canopy gap fraction is starting to decrease, and you can see also in the median values. And this is possibly indicating that the gaps are beginning to close as other plants are growing up to shade those areas out. But it's something that we need to continue looking at, because as the ash trees fall, they could also reopen those gaps and allow more light in. So this is looking at the relative growth rates of the native and invasive species, and this graph looks at each point represents the paired relative growth rate of one native and one invasive species. And the yellow circles represent the growth from 2008 to 2009, and the green triangles represent 2009 to 2010. And so you can kind of see if the plants were growing at the same rate, you would expect them to follow this one-to-one line. But you can see that the growth is kind of spread out. So in some cases, the native plant is growing faster than the invasive, but then also in some cases, the invasive plant is growing at a faster rate than the native. And so we need to tease this apart a little bit more. And these down here, where the invasive seems to be outcompeting the native, is where we need to focus our efforts, because that's where the invasive has the potential to take over. And we also are going to look at this data in a different way to see just the growth rate of an individual species as that changes over time, if it's growth released by the canopy gaps, if it suddenly starts growing faster, or how they might relate to different sites as well. So there's some more data analyses that we're going to do on this data, but it just kind of shows you that there is kind of a spread of growth rates at the moment. And just looking at what species that we're finding in these sites, so again on the top you see by the hydroclass, hydric, mesic, xeric, and non-ash, and there's a different number of plots per class, so you just keep that in mind. And of the total number of native species that we've counted, there's quite a few in the xeric sites and fewer in the hydric and mesic, and then the non-ash sites have even fewer, and those just seem to be kind of a typical of those sites where they are a little bit more of the mature forest, and so we're not seeing as much of an invasion present or just as much of an understory in general. And looking at the invasive species, of the 9 that we found in the sites, we found all 9 in the xeric sites, 7 of them in the mesic, 6 in the hydric, and only 3 different invasive species in the non-ash sites. So the most frequent species that we're finding in the understory is actually ash still, and the most frequent invasive species that we're finding are the common and glossy buckthorn and the oriental bittersweet. So this is looking at the information in a little bit of a different way. So here on the top we still have the hydroclasses, but going down we have the different species, including ash. We have ash and then the invasive species listed here, and you can see the number of plots that these are present in. So ash is present in 75% of the hydric sites, and 100% of the mesic, xeric, and non-ash sites have ash. And barberry is present in the hydric, mesic, and xeric sites, but not in the non-ash sites. And you can see Celastrus is really common in the xeric sites and less so in the hydric sites. So this kind of, if you go down the list, you can kind of get a feel for which species kind of group into which plots, which sites. And this can maybe inform different management decisions if you need to focus your efforts in a certain area or maybe a more sensitive habitat. So this is just looking at that data in that way and can be helpful for some situations. So to summarize, we have invasive species present in many of our sites, and it seems as though different species prefer different environments, which is pretty typical and can be useful for developing management strategies. And for the growth, we don't see a clear trend yet, but we also are interested in looking at different ways to analyze the data, comparing growth rates of the same plants over time, and also looking at it in relation to canopy gap fraction. So to kind of ask what's going to happen in these sites after EAB has gone through, it seems that ash regeneration is now dependent on the seedlings and saplings. Since we're not finding a persistent seed bank, and there's little to no new ash germination, those seedlings and saplings are what's really going to maintain that population. And if EAB continues to persist, it could pose a serious threat to ash in eastern North American forests. And in these sites, invasive species are present and have the potential to spread and may outcompete the native species in some of the situations and could be facilitated by canopy gaps. So basically at this point, time will tell, and we're looking to have one more field season up there where I will get more data and hopefully can have some more conclusive evidence afterwards for what's going to happen. So with that, I would like to acknowledge my collaborators in Michigan and in Ohio. A lot of the state and Metro Park staff were very helpful. We've also had research assistance from different personnel in the labs in OSU and MSU, and also our funding is from the USDA NRI competitive grants. So thank you very much, and I will take questions apparently after Kevin's talk. So I will turn it over to him.
Okay. I hope everyone can hear me. All right. So Wendy and I actually have very similar research interests. We're both interested in these canopy gaps that are formed due to EAB-induced tree mortality. And we're both interested in how the gaps alter ecological interactions and alter biochemical cycling and resource availability. However, I'm looking at it from a plant insect point of view. So the focus of my study is basically how these canopy gaps affect plant insect interactions in the forest understory. So to do that, I picked a plant called prickly ash. Unfortunately, the common name's a little confusion. It's not a Fraxinus species. It's actually a native citrus shrub in the family Rutaceae. And like I said, it's a native shrub, and it's typically in the understory. And this plant is really cool. It's got some really neat secondary metabolites called furanocoumarins, and these are extremely toxic compounds that are highly biocidal, and they deter most insect herbivores from consuming the foliage. Also, these furanocoumarins are phototoxins, so they actually increase in toxicity and concentration in the presence of UV light. So if you look at the picture there, you can see there's a prickly ash plant on the bottom, and this used to be an understory plant, and EAB came along, killed the ash tree that's above it. Ash tree dies, creating this canopy gap, and more UV light is hitting the foliage, and therefore my hypothesis is that this plant is now more toxic. Another neat thing about these the prickly ash plants is that it's dioecious, so there's separate male and female plants, and that sets up some cool theoretical studies I can do for plant defense. So females are supposedly, you would think, more defended than males. They allocate more resources towards defense or reproduction, where males allocate most of their resources towards growth. So although I said most insect herbivores can't consume prickly ash foliage, there is a specialist, the giant swallowtail caterpillar is a specialist on prickly ash in the Midwest. The picture is the butterfly adult is in the top, and it's actually one of the largest butterflies in North America. And the larva there, it's a bird drop mimic, and it's able to detoxify the pheranocoumarins with these cytochrome P450s. Basically, they can break down these toxic compounds. However, there's been a lot of studies that have shown even specialists, if you keep on raising the toxicity or the concentration of these compounds, there's a negative effect on the herbivore. And most of the time, it's a delayed development because the caterpillar's going to require more resources to go into detoxifying these toxic compounds instead of putting that resources into growth. And for something such as a caterpillar, delayed development is usually a very bad thing. You can pretty much say they're sessile organisms. They can't fly. They can't escape predators. So they're actually more prone to predators for a longer period of time, like birds, parasitoids, or even other insects. In the Midwest, they basically only feed on, for the most part, they only feed on prickly ash. And they have 2 generations per year, so it's a good study system for me. And they're ubiquitous in nature. They're very, very common. So we work in 7 parks in Michigan, and wherever I find prickly ash, if you search long enough, you'll find these caterpillars there. They're actually quite common. And last but not least, there's at least 3 recorded pupal parasitoids that are related, that actually attack the pupae of the giant swallowtail. So, just want to throw this map up. Everyone's probably seen this too many times, but where the red is, that's where EAB has been recorded, and I just wanted to show that our plots are basically right outside of Detroit, so they're in this very red area in Michigan, and that's important because all the ash there, like Wendy said, is basically dead. This is an aftermath forest. There's hardly any living ash there. So, to set up my experiment, my first summer here at Ohio State, I basically spent the summer hiking through the woods, and I was looking for prickly ash plants, and I would find, for instance, I found this male that's on the left here. It's tagged in orange. It's kind of hard to see. It's a male prickly ash plant, and you can see this used to be more than likely an understory plant. You can see all these dead ash all around it, and now these ash trees have fallen, and this plant is now in this large canopy gap, basically. So I would tag this plant, and then within 15 to 20 meters, I would locate another male prickly ash plant about the same size that was in an understory condition. So one that was surrounded basically by mature oaks and maples and hickories that it's going to stay shaded. So I would tag these trees, GPS them. I measured the length, the width, the height, and the DBH. And I did this for females as well. So I repeated this, finding a female in a gap and getting a shaded female nearby. So last summer, I actually got to do some exciting and actually collect some data. And basically, I have 2 parts that I try to connect here. I've got the plant physiology side, where I've measured the specific leaf area of the prickly ash. I measured the new growth. I measured the mechanical defenses, so I measured the number of thorns and the size of the thorns on the prickly ash. I recorded reproductive allocations, so the number of fruit on the females. And I'm currently working on carbon-nitrogen ratio, as well as using HPLC to quantify the pheranocoumarin and the total phenolics. On the entomology side of things, I did some feeding trials with the caterpillars to assess basically the relative growth rates, feeding them different foliage of the prickly ash, and I also did nutritional indices. For the sake of time today, I'm really just going to concentrate on specific leaf area and new growth and the relative growth rates. If you have any other questions about some of the other data, you can either ask me at the end or you can email me, and I'll be glad to answer that. So, specific leaf area. There was no difference between the sexes, but there was a strong, significant difference between foliage from prickly ash that was in the sun and foliage from prickly ash in the shade. And this is not surprising. This is what we would expect. This is basically showing that foliage in the sun, leaves in the sun are basically fatter, they're thicker, they have more cells in them, but it's important for what I'm trying to quantify, because they have more cells in them. Those cells are more likely to differentiate, and therefore I'm hypothesizing that they'll contain more phoranocoumarins. So I said I also measured new growth. So I basically went to each prickly ash plant and I randomly just marked 4 branches, and then I would measure the new growth. And there was no significant difference between sex or light. However, the p-value and the f-value there is that there was a significant interaction of light times sex interaction. So if you look on the left side of the graph here, and you look at the males on the orange bar, male prickly ash plants grew more when they were in the sun compared to shade. And again, that's not too surprising. That is what you would expect. But if you look at the female side, females actually grew more in the shade compared to females in the sun. And at first that's a little counterintuitive. You might wonder why. And my hypothesis is that the females in the sun are investing more in reproduction and defense compared to females in the shade. So on the Swallowtail Bioassay end, I went out and I collected first instar caterpillars from the parks. I brought those back to the lab and I maintained them on a diet of male prickly ash that was shaded, that I would think would not have very many of these phoranocoumarins on, so they're not pre-exposed to it. And they were randomly assigned, each caterpillar was randomly assigned to one of 4 treatments. Treatment number 1 is male foliage in the sun. Treatment number 2 is male foliage from the shade, and 3 is female foliage from the sun, and treatment number 4 is female foliage from the shade. So they're in these 4 categories, and when these insects basically, when the caterpillars molted into their 4th instar, I began the treatment of the bioassays. So I would measure the mass of the caterpillar upon emergence of the 4th instar, and I would measure the leaf area that I was feeding it from whichever treatment that it was assigned. And I ran these bioassays for 48 hours, and then at the end of it, I reweighed the caterpillar, and I analyzed the area of consumed leaf material, measured the frass, so I could get the relative growth rates and the nutritional indices calculations. So I did find that there was no significant difference in the sex of the plants, but there was a significant difference in sun and shaded plants. So if you look at this, caterpillars that were feeding on foliage from sunny prickly ash had a significant reduction in their relative growth rate. And the first thing most people realize when they look at that graph is, say, well, you know, that's barely significant. But the important thing to remember is that I only ran this trial for 48 hours. So these caterpillars actually have a life stage almost up to 20 days. So if you extrapolate this graph over their entire lifetime, I would expect that you would see a much more dramatic detrimental effect of feeding on prickly ash that's in the sun. So future studies, some of you might have noticed I had a very low sample size. It took me a while to figure out how to get these caterpillars to survive and transporting them. So this coming summer I'll be increasing my reps, and I'll be continuing the 4th instar bioassays, but I'm also going to include first instars in the bioassay treatment. There's been a lot of studies that have shown first instar caterpillars are often more prone to negative effects of secondary chemistry. Like I said, I'm also doing chemical analysis, and one of the most exciting parts that I think of the research is I'll be adding in this tritrophic interaction effect. So I mentioned earlier that there's several parasitoids that are recorded to be associated with the pupae, and I've got these pupae that some of them have overwintered. I got them to overwinter. You can see in the picture, and after the bioassays, I'll be releasing parasitoids and basically monitoring the survival and the clutch size and the sex ratio of the parasitoid. And so the goal here is to see whether the canopy gaps can basically cascade down to affect a third trophic level, basically, and possibly negatively affect it, or it could be positively affect the parasitoid. So again, I'm just going to talk quickly about a second side project that we started this past summer. And this project basically continues on and really looks at the changes in diversity and abundance due to the EAB, the changes in diversity and abundance of native arthropods. So this graph comes from the same paper, Gandhi and Herms, 2010 Biological Invasions. And they basically did an extensive literature search and recorded all arthropods that utilize ash as a host. And the first thing I want to say is there's a lot of species there you can see that use ash, but this is probably actually a underrepresentation because this is only recorded. This is only the ones that have been recorded on ash, and there's probably a lot more species that utilize ash, just hasn't been recorded. And they broke these species up into different categories from high risk to low risk species. So high risk species are ash specialists. They need ash, or they live their entire life on ash, and they require ash to basically reproduce. So you can see these ones might be high risk of local extinction due to EAB wiping out their food source. So one of the high risk arthropods is this banded ash clearwing borer. So this is a native arthropod. It doesn't cause damage like EAB, but it attacks ash. Typically does not kill them. And it's at high risk because of EAB is basically wiping out its food source. So to test some of this, we set up some pheromone traps for the banded ash clearwing borer. And one of the things I want to put out, a lot of people are like, well, why are you putting pheromone traps out for a species that may be in jeopardy? But the pheromones only attract males, so it's very unlikely that we're going to really upset the population if we're only collecting males in the traps. So I hung these traps up in the plots that Wendy worked in. So we've got this long history of data. The Cardina and the Herms lab have been working up there for almost more than a half decade now. We've got this large abundance of data of percent living ash and dead ash in these plots. And I basically hung the pheromone traps, and then I was going to do a correlation between the number of banded ash clearwing borers caught in the trap and the number of living ash in the transect. And unfortunately, I got no significant correlation, but I got to mention that this was a pilot study, basically, and we only set up basically 6 reps. And the 6 reps that we set up were in areas where there was high ash mortality in our plots. And I basically got hardly any of the banded ash clearwing borers in these traps. I would like to mention, however, that a few years ago we noticed some healthy ash in one of our metro parks in Michigan, Indian Springs. This is a picture I took of a healthy ash tree. It's the first healthy ash tree I've ever seen after moving to Ohio. And basically, we looked around and there's dead ash around this healthy one as well as other surviving ash. So there's several people at Michigan State University actually studying this right now. They're termed lingering ash, these ash that are staying alive. But I did hang pheromone traps there in this general area of this park. And at the time, we didn't have the percent living ash. We hadn't correlated that because it's not in our research plot. But I did catch 20 banded ash clearwing borers on average per trap. So it does look like there will be a correlation. We're going to repeat this experiment and also expand it basically and get the percent living ash and see if there's a correlation. So with that, I'd like to thank the following people with help with the research or the use of their parks and field assistance, and I would be happy to take any questions.
All right, we have both Wendy and Kevin here. If you have questions, please type them into the chat feature. Or what we can do is, if Kevin and Wendy, if you could type in your email addresses so that folks could get a hold of you after the presentation and talk with you, that would be good. I want to thank you too. Thank you very much for your information. I have a couple of questions. We've had some inquiries about as far as ash seedlings left over after EAB has gone through and killed most of the ash. Are the ash seedlings getting hit by emerald ash borer now, even the small ones?
Hi, can you hear me, Robin?
Yes.
Okay. So the question was, are these small seedlings getting hit by EAB at the moment? And that is, some of them are. So EAB attacks trees that are roughly 1-inch diameter. So as the seedlings and saplings get big enough to reach that susceptible size class, some of them are now getting attacked by EAB, which is the concern of, are these, is this orphaned cohort of the seedlings and saplings, is that going to prolong the invasion, or will it regenerate ash? But at the moment, it looks like it's actually prolonging the invasion because the EAB have a consistent food source as these saplings enter that size class. And we are still finding a low population of EAB even in these forests that have 99% ash mortality.
Okay. Another question, as far as the invasive plants that you're seeing, such as the buckthorns and the bittersweet, are these going to spread and become a problem quickly in these places that have been, where the ash has been pretty much taken out?
So the question is, are the invasive species gonna spread quickly? That's kind of dependent on the site. I know that some species do have the potential just for rapid spread, and a lot of the plots that we're working in just have dense, well, not a lot, but a good number of the plots that we're working in have really dense multiflora patches or honeysuckle patches, and so those seem to have this potential for rapid spread. Other ones, the privet, the burning bush, they don't seem to spread as fast, but it kind of depends on the site, where we see different species kind of grouping in different sites as well. So there's no clear answer for that yet, but it does have the potential for rapid spread.
All right. Thomas Munn would like to know, is there anywhere to find the listing of Fraxinus dependent species?
Okay, I would again point you to this paper, the Gandhi and Herms 2010 in Biological Invasions. They're really the only ones that have looked at this, and they mentioned they broke it up into guilds as well, and I'm not sure in the appendix if they list all the species. But this is the only available data is in this paper.
Okay, thanks, Kevin. And one more question for you. Is the surviving ash that you mentioned in the park a possible EAB-resistant variety? That's probably the question that many people have, is how did these survive and others not?
So the question was about the lingering ash?
Yes.
I'm sorry, I missed the first part. Okay. Yeah, so there's actually a big group down in Ohio State and Michigan State, I think, that are looking at the lingering ash, and if they are genetically resistant to it, or if they're just kind of the last ones to go. And so they're working on grafting and bioassays for that as well. So there's no answer yet if they could potentially breed a resistant cultivar or anything, but it's something that they are considering. And it looks like in some of their trials they are finding some success. And I know that they're also trying to cross native ash with the more resistant Asian ash, like the Manchurian. And so they are finding some success with that, but nothing official yet that I know of.
When you talk about them being EAB resistant, this means that they have not been attacked at all. Is that correct? The ones that you're looking, the lingering ash that you're seeing?
The lingering ash in Indian Springs do show signs of EAB attack. There's dead trees next to them, and I've actually climbed up into the trees. You can see bark splitting. You can see the D-shaped exit hole. So they have been attacked, and a lot of the ones, the picture I showed was the best example of a living ash. There's a lot that actually look like the canopy had died back initially, and that they've somehow sort of recovered after the attack. And we're hanging purple panel traps up there We don't know the extent of the population of EAB, but it does look, they are being attacked, yes, and it looks like some of them are surviving the attack.
That's interesting. Do you recommend planting native tree species in forests where ash is dominant?
So whether to plant native species, that's a good question. I know that some groups are planting native species to kind of, take over the gap before invasives have a chance. And I think Kathleen Knight is starting to look at that as well, with planting some down in Ohio across sites with varying levels of ash mortality. And it's possible that if you plant these species, then they could get established before the gap is really opening up, and then they could be there to take over. So it's possible. I don't have any definitive yes or no. I would refer you to Kathleen Knight for that question.
All right, thank you. I'm not seeing other questions. Are there more questions out there? And if so, why don't we have you please send your questions to Wendy and Kevin, who look like they've been doing a lot of work out there, and hopefully can give some answers to what Michigan is going to look, and Ohio and Indiana are going to look like here in a few years, if considering we're the states that are getting hit the hardest right now, or that we can see the most damage. So I want to appreciate all your hard work, and thank you very much for your presentations today. I'm sorry, there's one more question here. Any comment on hydrological impacts on the hydric sites?
So for the hydric sites, that's a good question, because those are where ash is more of a dominant canopy species. And I've seen some reports where as the ash die, the water table is affected. I think that's what the question is getting at. The water table is affected, and other tree species aren't as able to survive and remain in those sites. And we haven't seen enough of that yet, because the ash they've just died a few years ago, and it hasn't had that chance to really change the site as much. But I know that in some cases that is a big concern, that these hydric ash, black ash sites are just going to become kind of a marsh without trees. But I can't say that for sure in our sites yet. Sorry.
All right. Thank you very much. I will now bid Kevin and Wendy goodbye. Thank you very much for your work and your presentations. I'm going to put up a slide here where folks can email Amy Stone for credits for today's program. Hold on here just a second, and I will get that up for you. So here's some information for that, and with that, thank you very much for attending.